Thank you, Rene. It's a distinct pleasure to be here. And in the next 19 minutes or so, I'm going to talk about our efforts in the past few years to look at structure, function, and modulation of G protein coupled receptors. I'll give you a brief overview of what we have done in the last few years instead of telling you one story. I'll just take you through the research program in the next 20 minutes or so. So GPCRs, as most of you might have already heard about, they constitute the largest family of cell surface receptors in our body, and they recognize a diverse set of stimuli, hormones, peptides, lipids, and so on. And nearly every physiological process in our body involves a GPCR, whether we talk about vision, olfaction, cardiovascular regulation, immune response, and so on. And therefore, it is not surprising that about 1/3 of the currently prescribed medicines, they target GPCRs, and they essentially act by either activating or inhibiting one of these receptors. They continue to be one of the hot targets for designing novel therapeutics. As you can see here, over the years, they continue to top the chart in terms of number of drugs that are approved. And some of the examples of GPCR targeting drugs are listed here. Histamine antagonist for allergy, angiotensin receptor antagonist for hypertension, and so on. Even they have multiple roles in COVID pathophysiology. Angiotensin receptors, for example, they crosstalk with ACE2, which is the receptor for SARS-CoV-2. Bradykinin receptors being involved in bradykinin storm. Complement receptors critically involved in inflammation, and antibodies targeting complement receptors are currently in the clinical trial. And therefore, it's very important to understand how these receptors work, that is, how they are activated, how they signal, how they're regulated, not only to understand the fundamentals of cell signaling, but also to leverage this information in long run to design all therapeutics. Now, over the years, a lot of work has gone into understanding GPCR mechanisms, and I'll just take you through their classical, the current paradigm about their activation and signaling here. So receptor sitting in the membrane, when it binds an agonist is an activating ligand. Receptor undergoes the conformational change followed by coupling of heterotrimeric G proteins, hence the name G protein coupled receptors. This leads to the generation of second messengers, cyclic AMP, inositol phosphate, calcium, leading to G protein signaling. But this signaling has to be tightly regulated, and for that, cells have devised a mechanism referred to as receptor desensitization, where these GPCRs, they're phosphorylated primarily in their carboxyl terminus, which is inside the cell, by a family of kinases known as GPCR kinases or GRKs. And this leads to the interaction of a multifunctional protein called beta-arrestins. And you'll be hearing more about beta-arrestins in today's talk. And once beta-arrestins are bound to the receptor, they essentially come in the way of G protein coupling to the receptor, thereby blocking G protein coupling or terminating G protein coupling responses. This has been the classical paradigm. However, what has been discovered in the last 2 decades or so that beta-arrestins do much more than that. That is, they can mediate receptor endocytosis because they can nucleate various components of endocytotic machinery, such as clathrin, adaptin, and so on. And thereby, they can induce the trafficking of the receptors from the plasma membrane to endosomes. That is known as endocytosis or internalization. They can also scaffold multiple kinases and phosphatases to mediate parallel signaling pathway that is known as beta-arrestin signaling pathway. Now, the receptor G protein signaling is reasonably well understood through the work over past several decades, but receptor beta-arrestin interaction, the functional mechanism, is still emerging and remains in its infancy. And that is where our efforts have been focused in the past few years. Now, one of the approach that we use, I'll come to that. So beta-arrestins are essentially semi-symmetric bilobed structure. We have N domain and C domain, and they essentially consist of antiparallel beta strands. What is interesting is that the paradigms of beta-arrestin mediated receptor regulation and signaling is highly conserved across all GPCRs, and there are more than 800 receptors in our genome. But there are only 2 isoforms of beta-arrestins. That is beta-arrestin 1 and beta-arrestin 2. And our major thrust right now is to understand how these 2 isoforms of beta-arrestins are able to regulate such a large repertoire of GPCRs. As I said, the mechanisms of GPCR regulation through beta-arrestins and GPCR signaling through beta-arrestins appear to be conserved across this family. Now, one of the tools or approaches that we utilize in the lab is antibody fragments to probe receptor beta-arrestin interaction. And we utilize these antibody fragments or antibodies not only as biosensors to monitor GPCR beta-arrestin interaction, their conformations, but also to stabilize their complexes for structural analysis. We're all familiar with the conventional method of generating antibody fragments, that is hybridoma-based method, where we immunize animals with the antigen, followed by the generation of these hybridoma cell lines and isolation of the antibodies. This is quite cumbersome and time-consuming. What we use in the lab, what we have established as a platform in the lab is phage display-based library sorting, where we have a large collection of bacteriophages, that is viruses which can infect bacteria, and each of the bacteriophages essentially displaying one antibody fragment, either in the FAB format or in scFv format or nanobody format. And then essentially we incubate the library with our target of interest and then fish out specific antibodies against beta-arrestins, against GPCRs, against GPCR beta-arrestin complexes, and so on. And we have been able to isolate antibody fragments which are highly selective to beta-arrestin 1 isoform, for example, others which are highly selective to beta-arrestin 2 isoforms, other antibody fragments which can selectively look at or selectively recognize active conformation of beta-arrestin 1 or 2, and antibody fragments which can stabilize GPCR beta-arrestin complexes for structural analysis. And in the next few slides, I'll take you through how we have utilized these antibody fragments to look at critical aspects of GPCR beta-arrestin interaction and signaling and regulatory outcomes. So when we started the work in the lab, and before I go to that, let me mention here that because we are generating these antibody fragments from phage display libraries, we have their genetic blueprint, meaning we have their complete DNA sequence, and therefore we can simply put them in a plasmid vector to express them as recombinant antibodies in the cells that we refer to as intrabodies. So they're a very powerful tool because we can use them as biosensors of beta-arrestin activation, trafficking, even in live cells. So when we started working in this area, in the classical paradigm of GPCR beta-arrestin interaction, this was known to be biphasic. That is to say, in the first step, receptor here that is shown in red interacts with beta-arrestin through its carboxyl terminus. So the carboxyl terminus of the receptor that is shown here in yellow is the one that is getting phosphorylated. And this leads to formation of what we refer to as partially engaged complex, because beta-arrestin is engaged with the receptor only through the carboxyl tail. And then in the second step, beta-arrestin engages further with the receptor core or the transmembrane bundle, and it forms what we refer to as fully engaged complex. Now, in the field, it was generally believed that this fully engaged complex is the one that is functionally competent, meaning it mediates all different functions of beta-arrestins, that is desensitization, endocytosis, and signaling. But we quite did not buy into this idea, and we started to probe whether this partially engaged complex might also have some functional relevance. And using antibody fragments, we stabilize these two types of complexes in vitro and also in cellular context, that is partially engaged complex and fully engaged complex that is attached, where the beta-arrestin is attached only through the tail versus where the beta-arrestin is attached through the tail and the receptor core. And using a series of biochemical studies such as bioluminescence spectroscopy, we characterize that we have indeed stabilized partially and fully engaged complexes in vitro and in cellular context. And then we measured whether partially engaged complex of a GPCR beta-arrestin can mediate receptor endocytosis. And we are comparing here the endocytosis pattern of fully engaged and partially engaged complexes, and they're looking essentially identical. Similarly, in terms of signaling, we see that both partially engaged and fully engaged complexes of receptors and beta-arrestins are able to mediate ERK-MAP kinase activation, which is one of the key readouts for GPCR signaling. We have looked at this paradigm through using several different receptors, about a dozen different receptors, and this paradigm appears to be conserved. Therefore, Unlike what was conceived in the field and what was the general framework in the field, that this partially engaged complex has no functional relevance, we have been able to establish using several different receptor systems and a combination of different approaches that the partially engaged complex of GPCRs and beta-arrestins is indeed capable of mediating receptor endocytosis and signaling. And since we published these studies in a number of papers, several other groups have also essentially converged to the same conclusions using complementary approaches and different receptor systems. Now, going forward, as I mentioned earlier, there are two isoforms of beta-arrestins. One is beta-arrestin 1, the other one is beta-arrestin 2. And these two isoforms of beta-arrestins have about 78% sequence identity, and structurally also they're very, very similar. As you can see, these two structures, that is beta-arrestin 1 and beta-arrestin 2, are superimposed, and you can hardly tell the difference. But it is well known that even for a given receptor or different receptors, these two beta-arrestin isoforms are able to mediate distinct functions. For example, in case of a given receptor, beta-arrestin 1 may be critical for mediating desensitization of the receptor, while beta-arrestin 2 might be mediating endocytosis and signaling. And the mechanistic basis of how these two structurally very similar molecules, the two isoforms of beta-arrestins, are able to mediate distinct functional outcomes was not clear in the field. Again, we have stabilized and assembled these complexes of GPCRs with beta-arrestin 1 and GPCR beta-arrestin 2 complexes using antibody fragments either in vitro with purified proteins or inside the cell using these intrabodies that we have developed. And we have been able to demonstrate that the two isoforms of beta-arrestins, that is beta-arrestin 1 and beta-arrestin 2, they adopt very distinct conformations when they're bound even to the same GPCRs. And these distinct conformations of GPCR-bound beta-arrestin 1 and 2 are indeed driving their distinct functional outcomes in the context of signaling or regulation. We have further confirmed it by solving the crystal structure of beta-arrestin 2 in collaboration with Ha-Young Ho and Ka-Young Chung from South Korea, where we have crystallized beta-arrestin 2 in complex with the carboxyl terminus peptide of a GPCR. And here, this carboxyl terminus peptide is essentially phosphorylated, as it will phosphorylate in the context of the GPCRs inside the cells. And what we have seen here is that the structure of beta-arrestin 2 in red and the peptide is shown here in cyan. And when we compare the structure of this beta-arrestin 2, the first active structure of beta-arrestin 2, with the previously determined structure of beta-arrestin 1, the other isoform, we are able to see very distinct conformational changes between the two isoforms. The two domains of beta-arrestin 2, that is the N domain and C domain, they undergo a rotation of about 8 degrees upon activation of beta-arrestin 2, that is activation by this CXCR7 phosphopeptide, while in case of beta-arrestin 1, the domain rotation is close to 20 degrees. So the conformational signatures or conformational differences that we had seen for beta-arrestin isoforms using biophysical and cellular approaches are in fact further captured and thereby confirmed using X-ray crystallography approach here. Now, this was the structure that was done only with the peptide, that is the carboxyl terminus peptide of one of the receptors. And the next thing that we wanted to do is to capture the entire complex, because that is the real deal in cellular context. Peptide is just an isolated peptide corresponding to the C terminus of the receptor. The problem in capturing these entire receptor beta-arrestin complexes is that the receptor being membrane protein, it poses a very significant challenge in terms of structural characterization. But moreover, each of these GPCRs, they have multiple serine and threonines in their carboxyl terminus, which can get phosphorylated. And when you try to isolate homogeneously phosphorylated receptors, that is where all the sites or only specified sites are phosphorylated, it is very difficult. However, that is a critical step in capturing these complexes, because for structural analysis, the protein monodispersity homogeneity is critical. And therefore, using this vasopressin receptor as a model system, we have systematically characterized each of these serine and threonines that is present in the carboxyl terminus by mutating them one by one with alanine in alanine scanning mutagenesis type study. And then we have characterized to identify which of these sites which are present in the V2RC terminus are essential for beta-arrestin interaction. And we have been able to identify key sites, that is key serine and threonine residues, that are important for beta-arrestin binding. There are some sites, for example, this cluster TSS at the C-terminus end, which is dispensable. And this has allowed us to then generate a receptor mutant which has well-defined phosphosites, only 6 of them, and it can be used in turn to constitute or reconstitute a complex with beta-arrestin, and that is stabilized by an antibody fragment. So here we're using beta-1 adrenergic receptor as a model system. We reconstitute it in the lipid nanodisk. So first we express it in cultured SF9 cells, we isolate in detergent micelles, and then we put it in lipid nanodisk just to mimic the biliary environment. And then we complex it with purified beta-arrestin 1, and then stabilize the complex by an antibody fragment that selectively recognizes receptor beta-arrestin complex. And then we use this complex for determining the structure using cryo-electron microscopy. And this work is done in collaboration with Christopher Tate at LMB in Cambridge. As you can see here, we have been able to determine the structure of this receptor beta-arrestin complex at 3.2 angstrom resolution. And the receptor here is bound to formoterol. Formoterol is a long-acting beta agonist that is also used as one of the asthma therapeutics. Many of the inhalers that we use for asthma, they contain formoterol as the active ingredient. And the receptor is shown here in white. Formoterol is bound here in pink. Beta-arrestin here at the bottom of the receptor in yellow. And then the structure, the receptor beta-arrestin complex, is stabilized by this antibody fragment that we refer to as FAB30. So in absence of this antibody fragment, this receptor beta-arrestin complex is very, very transient, and it cannot be captured in a stable fashion to be visualized by cryo-EM or by crystallography. So the structure has revealed many key features of receptor beta-arrestin interaction. I'll just give you one aspect of it. So this is the receptor beta-arrestin complex, receptor in white and beta-arrestin in yellow. And if we compare it to receptor G protein complexes that have been determined before, if we superimpose them, what we see is that the G protein shown in G protein alpha subunit, shown here in red, and beta-arrestin, shown here in yellow, they sort of compete for similar binding interface on the receptor. Now, if you remember, I had said earlier that when beta-arrestins bind, they essentially occlude the binding site for G protein, which terminates G protein signaling. So this is the structural visualization of how beta-arrestin binding terminates or precludes further G protein coupling, because they're essentially competing for the same interface on the receptor. So essentially what we have observed in the last few years using a set of biophysical, biochemical, and structural approaches is that there is a great deal of structural and functional diversity in GPCR beta-arrestin complexes that drive their distinct functional outcomes and regulatory paradigms. Something that we did not, that I did not present today, hopefully next time, is that we have discovered a new class of receptors which we refer to as arrestin-coupled receptors. So these are native receptors present in our body that have very similar architecture to GPCRs, that is 7-trans membrane helix. However, they do not couple to G proteins at all. They couple only to beta-arrestins and they signal through beta-arrestins. So they are essentially naturally biased beta-arrestin coupled receptors. And we have characterized them extensively using signaling and structural approaches. And going forward, we are hoping to determine high resolution structures of multiple GPCR and ACR beta-arrestin complexes to visualize the diversity in structure and function at high resolution. We are establishing a SCRB supported National Cryo-EM facility at IIT Kanpur, which will be established as a national facility, and anybody can use it for structural purposes. So these are the people who have done all the work. I was basically talking about all the hard work that they have done. Each of them, they are very motivated, very sincere, and I'm absolutely blessed to have them in my lab. And we have been funded by Wellcome Trust, DBT India Alliance, DST, EMBO, DBT, CSIR, and IIT Kanpur. Thank you very much, and I'll be happy to take any questions.
Thank you for that lovely talk, Arun. I have a question for you. I work sometimes with insects, and I don't know whether you are aware that in insects, the C and N terminals are flipped. And, the C is outside and the N is inside. So people have, and that's only in insects. So people have wondered what could be the reason for this. And so do you, since you work so much in this area, I know it's a bit of an oddball question, but would you be able to give me an explanation for this?
So I think we do not understand why they're flipped, but what is known is that insect receptors are not in fact GPCR. So insect olfactory receptors, they do not couple to G proteins. While our olfactory receptors, for example, they indeed couple to G-olf family of G proteins. Why
So do you,
that is something that has to be looked at.
right, yes, that would be, but insects also have arrestants. So,
Yeah.
I guess it's fascinating, at least for me, to figure out, why we have this. And as you have also discovered a new set of receptors that don't seem to use G
Right.
proteins at all, but only use arrestants. So I guess we have a lot still to discover
Right.
about the
So
optimal.
in insects, we have only one isoform of arrestants. It's called KIRS. And one thing that I did not sort of bring up here, in addition to GPCRs, arrestants can also bind and regulate other classes of membrane proteins, transporters, ion channels, and so on. So perhaps some of the functions of this insect arrestant is for that as well.
Okay, fascinating. I must chat with you at, at length at a later stage. I believe that we are out of time, so I will thank you very much for that fascinating talk.